video, we'll be talking about protein structure and function. And within food science right now, there's a huge protein revolution where food companies are consistently asking questions about plant-based protein and protein functionality, and really getting to the point that consumers are demanding more protein and they're demanding more diversity in those sources. And so product developers are faced with a huge opportunity of formulation. Understanding protein chemistry really will help with understanding how to modify protein structure and function. So at the end of this video, you will be able to discuss the levels of protein structure, whether that's primary, secondary, tertiary, and quaternary. You'll be able to identify different types of amino acids and their functional groups. And from there, you'll be able to abstract about how those functional groups modify protein folding. Last but not least, you'll be able to discuss protein structure relative to some food structures. And so as I mentioned before, right now is absolutely a huge boom in protein chemistry because of the boom in diversification of dietary protein sources. I put this photo of a hamburger up here, but if we think about it from a systems-based approach, we've got protein within the burger. Is this a plant-based burger? Is this an animal-based protein burger? And if it's a plant-based protein burger, what are some of the different restructuring techniques that have gone into play to be able to mimic the structure and function of an animal protein-based burger? We've also got some cheese on there, and cheese is its own wonderful food source. I love cheese, and it deserves its own set of videos at a later point in time. We've got some sauces up here with where the proteins could be contributing emulsification characteristics, but protein has a lot of different roles in food from a structure and function perspective. Protein, for many foods, becomes the major macromolecule that contributes the structure of that food product. Proteins are enzymes as well, and in some foods the enzyme activity of that food is all related to the protein function. Protein contributes viscosity, it can be an emulsifier, and in some cases it's a color. I again am bringing up this burger as an illustration, but in one of the products that is out there, what is called the Impossible Burger, the coloration of that product is based off of a protein called leg hemoglobin, and it's the protein that I studied a long time ago in my PhD work. But it is a protein, and it is a protein contributing color specifically. So in protein, we have multiple layers of structure that we talk about, and in particular, we can put it into a sense of a hierarchy. That structure first starts with the 20 amino acids that are commonly found in biological systems. Now, there are more than 20 amino acids, but evolution, the way it's occurred, we really only see, for the most part, 20 amino acids showing up in plant and animal proteins. From that, we get a primary structure. And that more or less is the linear pattern of how those amino acids are put into order. Then we've got a secondary structure, how they start to fold in on themselves. Tertiary is how the protein as a whole starts to look. Quaternary structure doesn't happen for every single protein, but in some cases the proteins themselves will form different compound units where they group together in protein groupings, using some plain language here, to be able to form more biological systems or food systems. And we've got some great examples of quaternary structure being important for the function of foods. Last but not least, we denature proteins, and that's where we're completely collapsing these different protein structures on different levels. In some cases, we're just destroying the quaternary structure. In other cases, we're modifying the folding of the proteins at the tertiary and secondary levels. And in some cases, we're actually hydrolyzing proteins back to their primary structure and releasing free amino acids. So here's a visualization of this exact same thing that I was just describing. Primary structure, it's that chain of amino acids in a linear format. I almost describe it like the children's popping beads, where you make little beads into a necklace and you've got 20 colors, where those order could be any different order, but the coding for that goes back to the DNA within the organism that's expressing that protein. From there, those proteins start to fold in on themselves, and they're folding in on themselves based off of hydrogen bonding, based off of the charge density within the amino acids themselves, and how they are, the density. If you have a lot of charged amino acids in one area or not charged amino acids, they will tend to try and balance out as much as possible. Or how much hydrophobicity they have as well. Tertiary structure, again, how it all folds together to form the entire protein as a whole. And then quaternary, how they associate protein to protein to protein. So amino acids, these are the monomers. When I say monomer, I mean the singular unit that is found in a protein. And so these monomers, we use a very common nomenclature for these proteins, where commonly we call it an R side group here. That R side group could be, again, one of 20 different chemical compounds. Again, it's physically possible to have more than 20 compounds, but again, biologically, we really don't see more than 20. Some of those R groups are acidic, some of them are alkali, some of them are water soluble, some of them are not water soluble. And again, that variety allows for the enormous diversity of proteins that we see. So in the classes that I teach at my college, I don't expect people to memorize these, but depending on what school you're at, you may be expected to memorize the structures. What I'm expecting people in my class to get is that there's a huge diversity of different side chain structures, and that we see small ones, we see large bulky ones, we see ones that have these aromatic side chains, as they're known, which for the most part are very partly hydrophobic or very hydrophobic in the case of phenylalanine. We see the opportunity for charges on these proteins as well. And down here, we see this pKa value, and this is related to the pH at which the charge ratio changes from either being negatively charged or positively charged. You'll notice the ones that have positive charges, for the most part, have pKa ranges that are pushing slightly higher, and these ones are pushing down into the acidic range. I am throwing out a whole lot of different characterizations. Again, I teach applied food chemistry, and so I'm focused that people understand high-level topics and less on the minor details. But again, go back to what is required from you in your course. The students in my course specifically, I would like them to understand that there are these core topics. Amino acids could be uncharged. They could have a positive charge, a negative charge. They could be aliphatic or aromatic. Aromatic just meaning they have a large ring, or that they are hydrophobic but no large ring. Here's again some different examples. Again, I don't expect people are going to memorize this. What I focus on is the problem solving piece, but we need to understand the what behind problem solving before we can jump to the why and how of problem solving. So all of these amino acids form peptide bonds, and again, they all form this long linear process. I'm not going to go into the biochemistry of that, but in the DNA is the code for what proteins are going to be expressed, and within the ribosomes of the cell, those proteins are turned, or the code of the DNA is turned into an actual protein itself. Now, thinking about some of those amino acids, this is where we're going to jump into real food chemistry here. within those amino acids, there are opportunities for those amino acids to be interacting and modifying structure. One of the very common ones that we see in food systems is the formation of disulfide bonds. This is where cysteine amino acids, under the correct oxidizing conditions, can form an intramolecular or intermolecular link. And so if you've got two cysteines, when you create oxidation, those two cysteines can form a disulfide bond. And this is very, very critical, especially in the baking industry. And so, for example, in the case of gluten, we have two proteins, glutenin and gliadin, and they will form disulfide bonds under oxidizing conditions and hydration conditions. And those disulfide bonds are what allows for the elastic properties of gluten proteins. And so if we jump back here, think about the fact that it's cysteine that is bonding. In many food processors, if they do not want that gluten to form, maybe you're making biscuits or cookies where you do not want to have extensive gluten formation. Perhaps it's from a processing perspective, where the gluten would cause too much viscosity in your process, therefore you need to reduce the viscosity. Or perhaps it's all from an organoleptic perspective, where the gluten becomes tough and you don't want that toothsome aspect of your product. In many cases, they will add cysteine as a free amino acid, and that itself will bond with the gliadin protein and prevent gluten from forming. This is a very common food additive, and cysteine can be either synthesized through fermentation or it can be derived from a variety of different protein sources, but it's a common food additive, and cysteine is just an amino acid that's commonly found in food. And so when people come and say, but it's an additive, and I'm like, but it's already in your food, I always laugh. I'm a food chemist, always will be. We're on to secondary structure. Secondary structure is where we're starting to see those primary structure folding in on itself, and we typically see a few key motifs show up in protein structure. We've got alpha helices. The helices is the plural of helix, and this is where the proteins start to spiral in on themselves to make these coils. And we also see beta sheets. And this is where they're forming long linear strands and they pack into flat sheets. And how is this happening? Well, in some cases it has to do with the hydrophobic residues on those amino acids. The hydrophobic amino acids like to hide from water. Most proteins are found in aqueous systems. They're found in plants and in animals, and we are, to pardon my plagiarism of Star Trek here, but plants and animals are just ugly bags of mostly water. Our cells are mostly water. We are walking around water systems, and the proteins need to interact. The hydrophobic groups will hide and try to fold to within the interior of the protein. And so those hydrophobic amino acids, as much as physically possible, within the hindrance of that molecule, they will try and collapse on themselves. Similarly, if we've got a lot of positively charged amino acids, they will start to collapse in on themselves looking for negatively charged amino acids. But as we are starting to allude to product development, we can change the folding characteristics by changing the ionic species within the solution. So for example, if you were to add sodium chloride, you have the potential of modifying the charge density within the solution, and that can change the folding conformation of your protein. So start to think strategically about this. I also mentioned in that slide where I showed all the amino acids how changing the pH changes the charge density of certain amino acids depending on what pH you're at. Well, that's going to change the charge density of that protein as a whole, and it will change how it's going to fold or not fold and therefore change its solubility and its interaction within the food system. So your take-home message from the slide is this slide. If you can somehow open up that protein, you can get those hydrophobic amino acids out, and that's going to become important at a later point. If you can change the salt conditions in your protein solution, that's going to change your folding characteristics as well. Tertiary structure, well, this is where we're looking at the protein as a whole, where we're looking at what does it look like in a space-filling diagram. And so typically within food systems we have globular proteins. Globs, yes, globs. They look like balls. And so many of the insoluble proteins are going to be globular proteins. Again, typically a lot of insoluble amino acids tucked into the middle, and the polar amino acids on the outside interacting with the water interface. Some proteins naturally have a large random or disordered function, and these are proteins that are more soluble in water. They are more random configuration, and thinking about the association with water for proteins is going to be really important, especially in my follow-up slideshow. Last but not least, many proteins are fibrous in nature, and they're long linear structures where those fibers become critical parts of the biological structures, such as muscle fibers, where you have that linearity as part of the muscle structure, but is also at the molecular level. Last but not least, we've got quaternary structure. And again, this is where proteins are interacting with each other based off of charges or based off of hydrophobic residues on the surface of the protein. And those proteins can associate as dimers or trimers. In some cases, it's large networks of proteins, as in the case of gluten. And we see that interaction based off of hydrophobic interactions or charge-based interactions to form quaternary structures. One of our favorite proteins in food systems is hemoglobin, and this is where you've got a tetramer, 4 subunits of protein all grouped together. And what's also interesting about hemoglobin is that it contains a prosthetic group, not just amino acids, but it has an iron clathrate in a carbon-based ring structure that is associated in there. And that heme prosthetic group is what contributes the color to the protein itself. And again, hemoglobin is a tetramer, tetra being for the word 4. So, in summary, we have some core structures, and we've done a brief introduction into how we can start to modify structure with pH, with changing salt conditions. And the follow-up video for this is going to focus on a lot more of the functional attributes of changing the structure of proteins by modification of the biochemistry or modification of the physical chemistry to achieve the functional attributes that we're after. I like protein. I am always eager to talk to you. If you have questions, feel free to shoot them my way. Take care. We'll talk soon.